keep on with schedule, we'll move on to the final talk of the day. And guess what? That final talk is myself. So what I'm intended to do today as the last speaker is, we've been presenting a lot, well, my students have been presenting a lot lately about that metabolic status threshold. And I wanted to give a little bit of perspective about that metabolic impact and really to see where we came from and where we're going and how we are kind of incorporating this new data into our existing models. So as such, let me share my screen and there we go. Okay, laser pointer, everything is good. So basically what I want to talk about here, it's those photoperiodic versus metabolic triggers and how are they independent? Are they integrated? And where are we in our quest to actually try to clarify that? And then I realized that, in fact, I've been spending almost the last 20 years, 19 years to be exact, working on that particular topic. And it all started after I joined the University of Guelph in 2003, where my first interest was to identify some of the pieces, especially in the neuroendocrine part, so the hypothalamus pituitary area. And that will that helped us put together some connection. For example, we cloned the GnRH receptor on the pituitary that we could show was the driver to the response from the hypothalamus to trigger sexual maturation. And about the same time, Professor Tsutsui in Japan identified for the first time an inhibitory peptide that was the counterpart of GnRH to inhibit the sexual maturation or to inhibit reproduction. So all of those various pieces that we put together earlier on led us to our original model. And you can see there that on that original model published in 2009, there is no mention of metabolic status or metabolic threshold. In fact, this was exclusively based on our data and research regarding the photoperiodic control. And has been described a couple of times today, including by Clara earlier today, the main control of reproduction is that balance between stimulatory GnRH and inhibitory GnIH, a neuropeptide in the hypothalamus. And that hypothalamic in the medial-basal hypothalamus is actually here the integration center, which means this is where all of the external and internal factors are integrated by those neurons and then to decide whether to allow reproduction to proceed or not. And I'm not going to go into too much detail there, but basically what we originally showed was that the photoperiod controls the switch between inhibitory to stimulatory, meaning when you increase daylight, as Clara mentioned, you reduce the dark time, less melatonin, that decreases the inhibitory path, and at the same time, stimulation of deep brain photoreceptor leads to that increase in GnRH. So we've got here, the brake pedal is off, the accelerator pedal is full on. At the same time, we also characterize the receptors to this peptide in the pituitary, and we showed that as we switch from inhibitory to stimulatory, we also switch the sensitivity that further compound the activation of the reproductive axis. So from those, later on, we're still stuck, not stuck, but really focusing on exclusively looking at the photoperiodic control. And at the time, I acquired a genetically blind line of layers from Michigan State University, the Smoky Joe line that I've been maintaining at Guelph now for quite some time. And they were the perfect model to potentially look at the impact of light. Why? They have a degenerative retina, a genetic defect. So which means by 6 weeks of age, the affected birds have absolutely no visual perception. The retina is gone, so there is no visual input or retinal input. And using that, that strain of bird, that's also where we figured out that, in fact, the photoperiodic response has nothing to do with the eye and is located in those deep brain photoreceptor in the medial basal hypothalamus. And as Clara talked about this morning, we're expanding here to the turkey breeders, but the fact is red light is more efficient at penetrating the skull and the brain, and hence stimulate those deep brain photoreceptors. That led to the development of a commercial light bulb that is now installed in the many layer barns across Canada. However, while we were testing for those different type of light, incandescent, fluorescent, LED, and so on, we did observe the benefits of the red light, but something kept on bothering me all along while we were doing the studies. As you know, we have to pay to maintain birds. So what I was doing is mostly we were starting our treatment with birds that were already 14 weeks old. So we did not have to pay for maintaining those birds from hatch until 14 weeks of age. Problem is those birds are fed ad lib. And for whatever reason, in our research farm, they were always overweight, or they were always over target. And what we noticed while we were trying to look at our light treatment, what we noticed is those birds enter lay before they were photostimulated. And that was really kind of messing up with our data. So we looked a little bit deeper into it. And this is where we figured out, you know what, all those birds that seem to enter lay early, they seem to be overweight. Hence, in 2016, here we started to introduce that metabolic status in our model, saying that, okay, this is not only photoperiod, there is something inside the bird that tells it that it's ready to lay, and we really were looking at something linked to body weight. At the time, the leptin genes was just cloned in chicken, and of course that was one of our favorite candidates at the time due to its role in mediating the status of adipose in all the species. However, since we've kind of put the leptin a little bit on the back burner, as I'll explain in a couple slides. So that was then followed by a large, long experiment that Dr. Charlene Hanlon, who did a PhD on that project and is now a postdoc in my lab. That was a long study because we looked at 100 weeks into productions, and we grew the pullets. And what we noticed from our data is actually when we looked at the age at first egg, yes, there were differences. And what are those three lines? Here we've got in blue, a modern commercial line, a Lohmann LSL light. In green, the shaver heritage line that we maintain here at the University of Guelph. So here we've got a 2020 type of bird. In green, we've got a 2000 type of bird. And my Smoky Joe, a note here, we did not use the blind ones. We just used the sighted counterparts, but have never been selected for egg production. So hence they correspond to a white leghorn derivative of the 1960s. And we did observe that there was a difference at age at first egg. But what was striking is across those three strains, there was a very narrow body weight window where they laid their first egg, which really pointed out that, in fact, indeed, it looks like, yes, for those birds, we had three derivatives of white leghorns spanning, let's say 60 years of genetic selection, and still they all entered lay at the same body weight. So that kind of pushed us further towards that metabolic input. And as Mohamed just presented a little bit later on last year, that Mohamed presented that study, what we showed that, in fact, indeed, there was a metabolic threshold, but it's not set for all birds. And in fact, whites versus brown birds respond differently here to that metabolic threshold. So it's a kind of a sliding bar, I would say, that is dependent here on the strain, and probably here, as Mohamed showed, on body composition, because being the same weight, maybe you can compare apples and apples if we're comparing white leghorns versus white leghorns. But when we start to compare different strain, now it's not body weight, body composition is also going to come into play. And in addition to that strain and body composition side, as Mohamed showed, the environment, the rearing environment will also impact that body composition and will then impact body weight, and hence that metabolic threshold that we show was really narrow in our previous study. Here we see that it's still there, but it really depends on the strain, on the environment as well. So what are those markers? They're going to ask. So we're really now trying to focus on, okay, so can we identify specific mediators, just not body weight, body composition? And in fact, what we're doing now, we are really focusing on signaling, especially from the adipose. Well, the adipose is where the energy is stored, and that's where the energy is stored during the growth. And as Joanne and Thiago showed as well, that adipose accumulation is critical just in the prepubertal stage. It's really looked like around that time before the maturation. So before photostimulation, this is when we've got that increase in adipose deposition, hence pointing towards one of those key factors for the control. Now, as well, that's great to have energy in storage, but as well here, we need available energy. And we're also looking at potentially the impact of glucose metabolism, including insulin, for example, as part of it, that could also be mediators. And finally, the energy, well, either you use it to grow or you use it to reproduce. And in this case here, there is also then potentially an involvement of those somatotrophic axis, growth hormone, IGF, and to a certain extent corticosterone, which is not only a stress hormone, but which is also a metabolic regulator. So here you can see there we're looking at the storage, the availability, and the energy partitioning. And where would that signal go? Well, we are pretty sure we're still running some molecular analysis, but we are pretty sure that one of the main targets of some of those messages is the hypothalamic control. And that would be integrated, as I mentioned earlier in the presentation, at the level of the GnRH and GnIH neuron to try to balance that inhibition versus stimulation. However, some interesting facts have come up since we've done some studies, is that the hypothalamus is one thing, and that's to stimulate the overall axis, but there appears to be local regulation on the ovary. And for example, I'll just refer to you to this morning's presentation by Thiago, for example, looking at in broiler breeders, the impact on the ovulation process. And it really looks like that metabolic trigger also impacts the ovary. And in fact, we believe both should be coordinated for a successful reproduction. So just to give you a little hint, and just not to make it overly complicated, but here just to highlight a couple of key things that we have been looking at. So as I mentioned, GnRH and GnIH are the key ones that will regulate the overall activity of the reproductive axis. And interestingly, especially for GnIH, this is directly linked to appetite control. And in fact, GnIH, so during the growth phase, during the phase where the bird is immature, stimulate the orexigenic branch of the appetite control. And that leads here to an increase in feed intake. However, GnIH also inhibits the anorexigenic. On the other hand, here, when it comes to GnRH, GnRH is actually controlled by the orexigenic peptide. So as you can see here, we can really see that GnIH stimulate appetite that will go along with increasing the energy storage. But at the same time, as we increase appetite and we increase feed intake, eventually that could lead to an increase in GnRH. So we can really see this is extremely well coordinated. Now, on top of that, one thing we're really looking at, very interesting, is here the receptor to that adipokine called adiponectin. So the receptor adipo-R1, which has also been shown to be present in the hypothalamus, as well on both those orexigenic and anorexigenic neurons. So what binds adipo-R1? Well, adiponectin. And adiponectin here is the primary adipokine in chicken. In mammals, leptin is, but in the chicken, adiponectin is really the primary one produced by the adipose tissue. And as you can see here, I'm not going to go into too much detail, but we also included in that model the glucose metabolism, as well as some of the somatotrophic axis. Now, in the ovary, what is really happening is we need to initiate steroidogenesis, but as well, we need to initiate follicular growth. And basically, why those two needs to be coordinated? Because you need, obviously, to grow the small follicles towards yellow follicles and urachal follicles that will be ovulated. But we also need the expression of estradiol. And as I'll show in the next couple of slides, estradiol is very important to making that bird ready for a successful laying cycle. And if we're looking there, at the center of all that, what do we have again? We've got here adiponectin and another receptor, adipo-R1. So here we can really see that adiponectin right now is one of our kind of primary candidate as a potential mediator of metabolic status coming from the adipose tissue that can impact the hypothalamus and the control of reproduction, but can also impact here the ovary and the development and function of those growing follicles. So as I mentioned, why is estradiol important? Well, if we look at it, estradiol is at the cornerstone of the key aspect of egg laying. Number one, we need to have an oviduct that is matured for egg formation purposes. We also need to switch that liver from the standard lipoprotein, standard VLDLs, now to synthesize yolk-specific VLDL, and estrogen will be a primary effector on that. Those then yolk VLDLs can then be accumulated in the growing yolk. Estrogens also stimulate the calcium absorption in the gut, which means the hen knows it's time to interlay. When estradiol goes up, that means it's time to pack up calcium, increase the absorption, and as well, estradiol will switch the bone physiology from cortical bone to medullary bone. Good for egg production, but depending on the timing of that happening, if it happens too early, now you can see that hen is going to switch toward egg production, and that could come at the detriment of the musculoskeletal system, and as well could come at a cost in terms of liver metabolism by pushing for the overproductions of yolk lipoproteins. Now, when talk about skeletal health, Charlene just had a latest review, just accepted its impress in Frontiers of Physiology, focusing on the impact of estradiol. And here, as we can see, as we move from an immature stage to an inactive or to a mature stage with eggshell formation inactive or eggshell formation active, estradiol directly impact the function of the osteoclast and the osteoblast. And obviously, osteoclast, in this case, will resorb some of that medullary bone to release calcium, whereas the osteoblasts do the opposite. So hence here to show that there is a direct relation from estradiol on the bone physiology. And when it comes to the liver, as I mentioned, the same concept here. Without estradiol, the liver produces generic lipoproteins, but in the presence of estradiol, now we switch that liver and sends it into overproductions of those yolk lipoproteins for the purpose of egg production. That could technically and potentially lead to hemorrhagic liver syndrome as well, or fatty livers. So what is next? So as I mentioned, we've got our kind of suspects. So we have now a new toy at the University of Guelph. We recently acquired a multiplex platform to measure levels of messenger RNA in key tissues, and we've developed our panels, and we are like currently now all the samples we collected over the years, we're going to revisit those studies or those samples using that multiplex platform. As well, I'm not alone. as you know, Martin and I have been kind of playing with one another for quite some time now, and he brings his precision feeding system, which is an extra layer in control in terms of the nutrient delivered and nutrient intake. And finally here, a friend and colleague from Penn State University, Dr. Ramesh Ramachandran, who has its own assay to measure adipodectin, and is agreed to send me some of his antibodies so we can run those assays here in-house. And finally, we talk a lot about body composition. And I know we've run several DEXAs analysis, others, including the groups of Martin, has run DEXA analysis, and we know that it gives you good data, but there is not 100% accurate. And the reason why is the way the software is designed to analyze the scan results is based on mammals, and in particularly infant or small mammals. And guess what? These don't have feathers. And it turns out that the feather can actually potentially impact the interpretation of the scan results. And right now I've got a Sierra, a master student who is looking at here or developing a standard protocol for the use of DEXA, a more accurate protocol for the use of DEXA. And I don't know, I lost track of time. Okay, not too bad. And I would like to, of course, here thanks all the postdoc grad students and undergrad students that have worked with me over the last 19 years, because nothing would be possible without them. And as well, all our funding partners, CPRCs, EFC, EFO, CEFREF, NSERC, and OMAFRA, and I'm sure I missed some. For example, Alex Tees from Tees Electrical, when we were looking at the spectrum lighting. Okay, and I think this is it for me. So I'll be happy to take any questions if we have. So let me check the question. Okay. Okay, what can we learn from the literature on signaling pathway integrated with the activation of reproductive acids? Would you recommend investigating any specific metabolites from plasma? So that's an interesting question. And to be fair, on the layer side, there has been really not much done, because on the layer side, they were fed ad libitum, and really the people were just manipulating photoperiod. There is definitely more information or information on the broiler breeder sides that had to deal with the effect of overweight and the potential effect of obesity. Now, in terms of metabolite, I think the adipose, from the adipose, and one thing that I did not mention actually is potentially the difference in free fatty acid. Because if in some of the graph that I presented there, I kind of had that PPAR gamma, and PPAR gamma is a nuclear receptor which seems to be really implicated at every level. And guess what? Some of those free fatty acids are ligand for PPAR gamma. So that would be I think that would be really interesting to look at the contributions of different types of free fatty acid, and could they actually skew or help with that metabolic control, if you ask me. And as well, of course, I think ultimately glucose is important. That's the energy source or the fuel for the body. But in metabolites, I'm really interested, and if you're interested in doing that, it's kind of looking at the impact of some of the free fatty acids. And in terms of readings, you know, I don't want to always say, oh, you should promote my own students, but Charlene Hadlon, over the last 2 or 3 years, has put out a couple of excellent reviews, in particular, Should I Lay or Should I Grow? It's in Frontiers. I would highly recommend to read that one as a starting point, definitely. All right. Is there any other question? Okay, well, if there is no other questions, I guess that's kinds of wrap ups today's meeting. Great day. As you can see, there is a lot of great research going on, and there is a lot of complementary research which is done across Canada, especially between institutions. And even if we're not necessarily collaborating, hopefully that CPRF platform could serve as a great starting point for further collaboration in the future. And again, I would like to thank all the sponsors for making this possible. And as a closing remark, well, I'm looking forward to see you all tomorrow morning for our last day of this event. And again, thanks to all the speakers and all, of course, all the attendees for today. All right, great. Thank you very much.